I'd like to say hello and welcome to everyone in the Wolfram Mathematica community. My name is Alan White, and my topic is the kinetics of the prostate antigen. The equations I'm going to present are almost all due to Ernest Rutherford, so I'm going to do my best to live up to his motto, which is to seek the first principles of things. Now, this is the current state of affairs between myself and the clinical community. I would like clinicians to take their time and concentration numbers for the PSA molecule and curve fit them to the logistic equation. And right now, they're perfectly happy with linear approximations, which is PSA velocities and PSA doubling times. Now, here is an image that kind of got me started. This is antique. It's all the way back to the 60s. And the artist here is describing prostate tumor cells, and he's showing 5 different levels or grades, which I would say that's discrete behavior. So he's already talking quantum mechanics. And what I'm hoping is that each one of these grades can be fit to a particular logistic equation with a certain time coefficient and an increment. And of course, we have to exclude the other equations. Now, let's suppose you're a teacher, you have students. This is the goodness in my book. I have manipulates for all of you. All you have to do is work the slider bars, and you can simulate any patient encounter that you wish. You don't have to rush to a hospital and ask for laboratory data. Now, let's suppose you do it that way. You move the slider bars, and you have the numbers that you like. You scroll down in my notebook, and you find that first equation, so you know where you are. The second equation, I've tabled the function for you. So then all you do is push Shift-Enter, and you've got a set of data points. Okay, now what do you do with the data points? Well, you can send your student to the Memorial Sloan Kettering webpage, and they can calculate doubling times and velocities, or you can do it yourself. Another very good place to go if you want to find a clinician to work with is visit the Urology Today webpage, and you'll find any number of videos where very intelligent state-of-the-art physicians are holding conversations. Okay, now this is the diagram that really is the fundamental start and finish of my work. What we're going to talk about first is postoperative patients. Now with these guys, what happens is the surgeon goes in, he takes out the prostate gland and some surrounding tissues. Hopefully he gets it all. And if he does, there's a darn good chance that the PSA molecule will disappear from the patient's circulation following the A trajectory. Now in my notebook for this, obviously, every patient is going to have a different starting concentration. So what you need to do is you normalize to compare one patient to the next. You have to normalize the numbers. And of course, with this equation, the only variable really is the time coefficient. Now, this is something special with my interpretation of the logistic equation. I made a study of Roberval's work a great many years ago and came to realize that for the bounding rectangle to be in correct proportions, the aspect ratio has to be not the golden ratio, but 2 divided by pi. And when you do it that way, a stable equilibrium will pass through the center of the bounding rectangle. That's point P1. Now here's our mentor, Professor Verhulst, and you all know he got into conversations with Professor Quetelet, and they decided the world needed an equation that would describe population growth from above. So that's where we are. That's how we got it. Now, here's my manipulate for his equation. The green line is basically stable because it's going right through the center of the bounding rectangle. And I set up 3 patient simulations. You can see the variables of interest are the time coefficient, the increment, and the initial concentration. Now, obviously, it's not going to be that simple. So what can happen? Well, if you have a patient who's lucky enough to have pure exponential depletion, I should do it that way for you. If he has pure exponential depletion, the p-value for the time coefficient could be as high as 10 to the minus 20. If you have a mixture distribution, on the other hand, if this guy is half logistic and half exponential, the p-values get a lot worse. So that's how you can sort out. You can tell if your patient has a pure pathway or if it's a mixture pathway. Another issue is each pathway should have its own bell curve. So obviously, if you have a fast rate of descent, you're good to go, maybe. So you're in the green half of the bell curve. And if things are going slow, well, then you're somebody to worry about. But there might be more than one bell curve. So I'm insisting to the clinical community that they need to run tests on thousands and thousands and thousands of patients because there's issues having to do with distribution models. And we need to know if we need a distribution model. Now, another thing, each equation that is in my book, I set up a first and second derivative. So here's the inflection point. And as you can see, it probably is a stable depletion because the acceleration points, max and min, are sort of like on bordering on both sides. Okay, now let me give you a very powerful analogy here that'll help you understand why I'm insisting. There's the PSA molecule, very complicated, all sorts of amino acids. It's stuck onto the tumor cell membrane. Anything can happen there in the membrane. You know that. I don't have to tell you that. Now, let's think for a minute about the AIDS virus infection. You get the AIDS particle into your bloodstream. If you have a CCR5 receptor, the AIDS virus can get into your cells and you're in trouble. If you have a 32 base pair deletion out of your genome with zygosity, there's a chance that the particle will not be able to make it into your white cells and you're immune from the disease. And my thinking is there might be similar subtle issues. You know, only 10% of the population is resistant to an AIDS infection. So I'm thinking the PSA molecule, it could be just as subtle. All right. That's surgical therapy. Now, what about radiotherapy? Well, what happens then is you're laying on a table and there's this device up there and it focuses an energy field and hopefully it'll hit your prostate gland. Well, if that happens, then the PSA molecule concentration should follow the B curve by my light. And here's my Mathematica notebook for you. The blue rectangle, that's the period where you're getting the therapy. And I've got 3 patients. So obviously, you want to be purple dots or blue dots. You don't want to be Mr. Red Dots. And I set up an integrate so you can calculate surface areas under the curve and also find maximum and find minimum. And you can see there's his equation has basically 2 time coefficients. Now, here's my thinking. I tried to explain to you about washouts, logistic and mono-exponential. I think what you do is after you calculate a find maximum, you use that as the first point and you take the rest of the downdrifting points, put them together, and then you try to curve fit to mono-exponential depletion or logistic depletion. And I would say intuitively, I would rather be Mr. Mono than Mr. Logistic. But that's but you see there's an infinite number. Graduate students are going to run with this. I'm sure, I hope you will agree. Now, what is brachytherapy? The surgeon goes in and he has some pellets that are radioactive and he implants them around the perimeter of the prostate gland. So you always have the tumor, but hopefully you're going to get rid of all the dangerous cells. And that, I think, by my light, is Rutherford's graph C. So I tried to set up Rutherford's graph, and what I found is he had 3 time coefficients, and I couldn't get the curve to drop down. So I simplified his equation with 2 time coefficients, and that's Mr. Purple Dots. He's good to go. You can see it went below the threshold values. So clinicians say cutoff points. Now, let me see if I can try and explain this to you. For whatever reason, Mr. Red Dots and Mr. Blue Dots didn't do well, even if they got the exact same dose and time frequencies, they still have malignant cells in their high energy columns. So they're at risk to the tumor growing and also metastasizing. Mr. Purple Dots came out lucky. For some reason, for one reason or another, Mr. Purple Dots, we ablated all of his aggressive cells, and hopefully, because he's surrounded by the radiotherapy, they might not come back. Now, getting back to my interpretation of the logistic equation, think about your data points going through the inflection point. If they pass through the inflection point, what that means is there's no new mutations. If they go above the inflection point, you're in trouble. It's probably the tumor's getting hotter. And if your data points go beneath the inflection point, there's a good chance that you're better off. Now, here's another curiosity. For some reason, radiotherapy patients, years after the therapy, the PSA returns for mysterious reasons. Clinicians have this down. They're saying, well, if it's 2 years or less, it's a harmless bounce. And if it's out there at 4 years, it might be something to worry about. Now, my thinking along these lines is let's try to curve fit the data points to de Moivre's equation. And I would think if it's a harmless bounce, it will fit to de Moivre's, and we'll get these metrics for the first and second derivatives. And here's the first and second derivatives for a logistic encounter. So you can see there's quite an obvious difference. So ignoring the calendar, put the calendar off to one side, you can see it's possible that using these equations, you'll be able to sort these patients out. Now, again, the issue is how soon can you tell the clinician that he's good to go? Well, here's 5 years of curve fitting on the top. You can see that's nonlinear model fit, which you all know, so it's perfect. Here's beneath that, there's a couple of years of data points, so the fit's not quite so good. But here's what the clinicians have right now. They're just using linear approximations. So I think they would be better off doing it my way. Now, I've hinted that a binomial distribution will describe everything. So let me try to explain to you how you can get into the malignant state. In the upper left-hand corner, you've got a nodule or a lobule of cells, and there's 10 genes running the shop, and the skew isn't too high. It's 0.525. So you got about 1,000 cells and about 7.5% in the high energy level. Now, if you increase the skew, take the skew up 10% or so, what happens is the number of cells in the high energy columns jump up to almost 32%. So they go up almost by a factor of 4. So now you're in a state where you have a cancer for sure, but maybe it's a low malignancy tumor. Now, there's another pathway to malignant behavior, which is worse. What can happen is genes can drop out of your ensemble. So you can go from 10 genes down to 7 genes. When that happens, your tumor lobules shrink down enormously. They get much smaller, see, from 1,000 cells to 128. But instead of just 32% of the cells in the high energy columns, you're all the way up around 60%. And I should say, parenthetically, as an aside, I was a pathologist for 25 years and I saw all these things. So I'm not making it up. Now, here's something that if it ends up where you like this and you end up talking to clinicians, a radiotherapist is going to tell you, boy, healthy cells, they just resist that radiotherapy. And those cancer cells, we get rid of them when we irradiate them. Well, I can explain that too with a binomial. You see, here up on the top, I've got a bunch of sort of like intermediate aggressive cells. They're mostly askew. They've got 10 genes, but the skew is up. So when you irradiate them, the cell concentration goes down by a factor of half. You go from 1,000 cells almost down to 500. Where here's a healthy nodule of cells. So what happens in this case is after you get rid of all the cells in a high energy column, you only lose about 10% of the total cells. So you see, clinicians they'll share anecdotes with you because they want your help. And the anecdotes lead to a binomial explanation for all these things. Okay, now here's Donald Gleason. He's the surgeon who figured it all out for prostate cancer. And he said the cell populations are discrete. There's type 3 cells, see, and he's calling 3 plus 3. And then he says there's 3 plus 4s. So which is not so bad. But then you can have 4s plus 3s, where the 4s are a lot more dangerous than the 3s. So that's what you don't want. So Donald Gleason, by my light, he's the Max Planck of the microscope. And Max Planck, he says the law of distribution in the normal spectrum. And I think Planck is basically saying that there's a binomial distribution there somewhere. Now, I just showed you, I was explaining a mixture distribution, Gleason's 3s, Gleason's 4s. Well, in my book, you have that all there for you. I've got bar charts set up so you can use, and I also have continuous distributions. Now, let's think about a Gleason's 3 plus a Gleason's 4 mixture. Your Gleason's 3 cells are going to have 10 genes working in the ensemble. The skew is going to be too high. So about 26% of the cells maybe, will be in the high energy columns. Now, the Gleason's 4 cells, the really ugly looking ones, they're only going to have maybe 8 genes functioning in the cells, and they're going to have an accelerated skew. So you're there over 50% of your cells in the dangerous high energy columns. Now, am I seeing, would this make sense to a clinician? No, it won't. But here's a cartoon that a clinician made. And what they're saying is there's 2 subpopulations in a cancer cell tumor. In a cancer tumor, there's high energy cells and low energy cells. And when they give either radiotherapy or chemotherapy, they're destroying the high energy cells. See, so they're seeing it exactly the way I see it, but they don't want to go to any level of abstraction. Now, certain of you with math PhDs, you're not swallowing this because you're saying, why does he think he can go back and forth from continuous to discrete? And I would say to you, you didn't study Abraham de Moivre's equation. De Moivre's equation draws a smooth curve with the same independent variables, n and p, as the binomial distribution. So that's why I'm comfortable with this idea. I see a smooth distribution in the microscope, but I'm able to picture a binomial distribution because I'm familiar with the work of Abraham de Moivre. Now, this kind of summarizes the work I did with black body radiation, because I think black body radiation is the beginning of everything. So there's Planck's equation, and then the blue rectangle is the visible range. Now, a few years later, Albert Einstein came along in 1917, and Einstein came up with an equation slightly different from Planck's, and I tabled it here. Now, the point I'm trying to make, see, there are 7 columns in the visible range because Isaac Newton told us that the visible range has to be an octave. So there has to be 7 columns. If there's 7 columns in the visible range, then give or take, there's about 42 columns over the entire range. Follow me? Okay, it's just addition and subtraction. So what you have over here on the far right-hand side, you have long waves, and then you have the visible range, and then the green columns, I would say, are electrons, and the red columns, that's where your photons are hiding. Now, that diagram in the upper right-hand corner, that's from the book Planck published in 1914, and he's saying things like distribution density, it's changing abruptly from one region element to the next. So he's saying there's columns, but he's calling them region elements, you see? And here's what Einstein said a few years later. He's saying the same thing, discrete set of states, statistical, mechanical, blah, blah, blah. And then what does he do? What does Einstein do? He uses Boltzmann's equation for single-sided probabilities. So I'm picturing Albert sitting at his table, and he's got Planck's book open, and he sees this one-sided chart here. So that's why he called on Boltzmann's equation. Now here again, this is what you're going to find in the clinical literature. You're going to find casual applications and statements of the logistic equation. Okay. Now, this is my idea about the way radiotherapy works. The patient jumps on the table every day, Monday through Friday, and he gets irradiated. And if everything works well, let's assume there's like a 20% ablation rate, you know, all those cells in the red columns, each time he jumps on the table. So at the end of 30 days, you can see the tumor population is drastically reduced. There's still a few cells left, and the problem is that they're non-binary. Tumor cells don't have to mate in order to duplicate. So here's the point I'm trying to get across. This guy, the guy who's going to get a cure, each time he gets on the table, all of the high energy cells are ablated. So cells from the green energy columns are going to migrate into the red columns, but the next day, Powie, they get it. You know, everything works out for him. The timing is just right. For a guy who's not doing so well, for some reason, the next day he's got a lot of cells left over in the high energy columns, and those can duplicate and metastasize. Now, I know you're thinking now that I painted myself in a corner, because if tumor cells are non-binary and there's always a few cells left, how do you get a cure? Well, here's an interesting paper that was published in 2019. And what the authors did is they took 2 groups of prostate cancer guys, and they irradiated one group for 28 days and the other group for 41 days, but they gave the total dose the same, and they got the same cure rate. So you see, what would happen by my idea of 20% a day, there'd be over almost 4,000 cells for the one population left over, but only 275 for the other population. So how do I resolve that paradox? It's not too healthy looking. So what I want to do is invoke area equations. What I'm thinking is, you irradiate here, you know, on day 6, -6, the cell death rate goes up. That's the blue line. You irradiate here on day -2, and the cell death rate goes up so high that you get a cure. So in other words, my statement to you is, when you cure cancer with radiotherapy, you have achieved a chaos. You have brought about a state of chaos. Now, what I'm hoping some years down the road is that graduate students are going to approach me, and they're going to have solutions to my interpretation of the logistic equation. And those solutions are going to have, each Gleason's grade is going to have a different time coefficient and a different increment. It's also my hope that this is the 5 hours of radiotherapy that the clinicians will talk to you about. And I'm hoping that we can come up with more rigorous definitions, more abstract reasoning. Now, let me explain one of the variables here, because you're probably saying, it doesn't make sense. One guy cured, the other guy not cured. What you have to think about is double strand breaks. The way you destroy a tumor is you bring an energy field into the tumor and you inflict double strand breaks on the tumor. And when you do that in a DNA molecule, that's what destroys the functioning of the cell. But, ladies and gentlemen, going from one guy to the next, every guy in this room has a different concentration of base pairs that predispose to double strand breaks. And if I recall correctly, I think it's a CG step. So if you have a lot of C's and then a G and then an A and then a T, and you have a lot of C's and G's, you're more vulnerable to double strand breaks than the guy who has fewer CG base pair steps. So you see, there are subtle issues like that that have to be winnowed out and they have to be identified. If it was up to me, every time somebody was biopsied before their radiotherapy, I would sequence the genome of the tumor tissue, and I would say, how many CG steps are there? How vulnerable is this guy to radiotherapy compared to another guy? And that's not being done just yet. That's an idea that's kind of nascent. But you see how much room there is for you. This is not a project that I'm going to accomplish by myself. This is something where I have a book, and hopefully graduate students will get their hands on it and they'll run with it. So now for my last slide, if you're going to ask me questions about statistics, I'm going to plead an Isaac Newton, and I'm going to say, go see Mr. DeMovry. And I also want to say, you all have math PhDs probably, so I'm also very pleased to be in a room with guys like that. And see what Ostwald said in 1904. He said, I see among my audience men who are incomparably better educated than I am. And there's a lot of happiness being able to say something like that. So that's where I am. And if you have any questions, we've got plenty of time. So please go ahead. The way I got this idea, I originally wrote a manifesto. When I found this diagram, I wrote a manifesto, and I was basically saying to the mathematical biologists, it's a mistake not to use all of Rutherford's equations. Then I read a paper on prostate cancer, and the author said that during radiotherapy, the concentration zooms up and then it comes down. But yes, the PSA molecule will follow course B. And so it does change. So it is giving you some indication of whether or not how effective your therapy is, because you want it to you want the molecule to disappear completely. See, this is the manipulate that I set up. And what I'm showing here is 3 patients, 3 guys. They're all making it beneath the cutoff points. See, that's what clinicians have now. They don't use fine minimum. What they use is they establish arbitrary cutoff points, and they watch to see the concentration go down. And then they use a ruler and a number 2 pencil, and they draw a line. See, and that's the practice of medicine. See, so like I'm saying, yes, you're right. The PSA concentration does follow it. Now, how closely? Well, I can't tell you because nobody thinks like this. I think like this. It kind of makes sense to you. You're a little bit skeptical, but it kind of makes sense to you. But what I've been doing is I've been trying to crowdsource my book and get it published. And what I'm hoping is they will take up on that. But I don't know how often to draw a specimen. I don't know if you need a specimen every day or every other day, because you're going to be laying on that table for a whole month. See, but see, the clinicians want to go golfing on Saturday and Sunday. So what they do is they radiate Monday through Friday, and then they say, well, let's give the guy Saturday and Sunday off, But it's possible that between Saturday and Sunday, the tumor population grows so high that it starts to metastasize. But the hidden variable there is, do you have CG steps in your DNA molecules, which makes your cancer vulnerable? Or are there no steps there, so you're resistant? And that's not worked out yet. They're just starting to see things like that. Have I answered your question? I do not know if a clinician would take a chance on changing a patient's individual schedule in real time. What would have to happen is they would have to do hundreds of patients and then go have a big national meeting and say, you know, how are we doing getting the specimens at the right frequency? So what you're saying is, well, if they keep an eye on things, can they jiggle the handle while the guy's on the table? And the answer is no. I don't think that'll ever happen. But if you have hundreds of data, like I said, what you want to do, if you like this, go to the Urology Today webpage and watch the videos, and you'll pick up on their lingo, and you'll understand. And they also, at the webpage, they list all their papers, so you can get to their papers for free, most of them. And they talk about, well, here we had 1,000 patients here, we had 200 patients there. But I see, that's what I was trying to show, that I set up, you can do sample calculations as a graduate student. You don't have to go into a hospital and get numbers. You can sample calculate, then you take them to the urologist, and you can say, you know, does this make sense? But your idea that you would adjust during the therapy, that's not going to be anytime soon.